This protocol allows us to determine whether missense variants in human genes that are linked to diseases affect protein function. This is difficult to accomplish computationally, even with state-of-the-art algorithms. This technique allows a rapid analysis of missense variants in human proteins using an in vivo system Drosophila melanogaster, which is faster and more cost-efficient than vertebrate model organisms.
Demonstrating that a specific variant affects protein function directly contributes to the diagnosis of rare diseases and serves as a starting point for disease mechanism analysis and potential therapies. This protocol is particularly useful when studying variants linked to rare diseases, but it can be applied to more common diseases like autism and cancer.
Identifying reproducible phenotypes in flies that reflect protein function can be challenging. The phenotypes may vary from gene to gene and can be subtle.
Functional studies of variants in human proteins using Drosophila can be performed based on rescue experiments or overexpression studies. Before beginning the procedure, gather information about the human gene of interest and its model organism orthologs. To test variant function by overexpressing reference and variant human proteins in the fly visual system, generate transgenic flies that express reference or variant human cDNAs of interest under the control of the Gal4 upstream activating sequence, or UAS, system. To select a specific Gal4 line to express the human proteins in a specific fly tissue, cross 3 to 5 virgin females from the Gal4 line with 3 to 5 males from each of the UAS human cDNA transgenic lines in single vials or in duplicates. Transfer the crosses every 2 to 3 days to obtain as many animals eclosing from a single cross as possible.